I did hand this out yesterday. So both processes have Okay, so one of the questions in the quick check for the 8.5, I'm asked you to compare and contrast these, so I thought that'd be a good thing to talk about here at the beginning of class. So the first two questions are similar about the two processes. The 3 and 4 are differences. So number one, what's the energy from the movement of electrons used to do? And again, so these yellow arrows here are showing the movement of electrons through the electron transport chains. The energy from the movement of those electrons is used to do what? Yeah, Lucas. Yep, to pump the hydrogen ions across the concentration gradient. So and here you can see the hydrogen ions are getting pumped into the intermembrane space in the mitochondria. Here they're getting pumped into the thylakoid space in the thylakoid. Okay, so that's what this movement of electrons, the energy of that movement is used to do. So number 2, why do we create that hydrogen ion gradient? What's the purpose of that? Yeah, Richard. It's used to make ATP. So when the hydrogen diffuses back out from high to low, it goes through ATP synthase, and that ATP synthase is gonna make our ATP. So it's gonna add that phosphate group back to ADP, make our ATP. Number 3, this is the difference between the two. What molecule donates electrons in cellular respiration? It's our NADH, our electron carrier. And those are very high energy, so they're going to get pulled all along on their own. In the light dependent stage of photosynthesis, that donator is water. Okay, and those electrons need a push. And what energy is providing that push? The energy from the sunlight, right? Chlorophyll is in these photosystem proteins here, and it's absorbing that sunlight and causing those electrons to be pushed along. Number 4, what molecule takes the electrons at the end? In cellular respiration, it's the oxygen, right? So that's why we form the water, because the oxygen is accepting the electron. And in photosynthesis here, at the end of the electron chain, what's picking those electrons up? Yeah, our electron carrier, our NADP plus, is picking it up. And now it's reduced. It has those electrons and has those hydrogen ions. Okay. So yeah, Richard.
Number 4, it was NADH and water.
So technically, it's oxygen that's picking it up, and the result of that is water for cellular respiration. And then in photosynthesis, the NADP plus is picking up the electrons and getting reduced to NADPH. Does that make sense? Oh, sorry.
I'll toss it back.
Oh, number 3, I'm sorry. So yeah, in number 3, the NADH, yes, I'm sorry. NADH drops it off in the cellular respiration and water, the electrons come from water and photosynthesis. Yes. Sorry about that. So any other questions on those processes before we move on today? Yeah. So we haven't really gotten to the Calvin cycle yet, but it's the second part of photosynthesis. That's what we're going to talk more about today. Yeah, I guess another important difference to note is that this is the end part of cellular respiration, right? So that ATP is then going to get used for the cell for any process that it needs to. In photosynthesis, and not all of the ATP has to go to the Calvin cycle, but generally a large amount of that ATP ends up going on to the Calvin cycle and not to the rest of the cell for other cell processes. Okay, so that's another difference. Okay. All right. So, like I mentioned, we're going to now talk about the second part of photosynthesis. So, I know this is a little low for some of you, but we looked at this intro diagram yesterday, and this will be in your second quick check today. We talked about the light reactions yesterday. We're now going to talk about the Calvin cycle today. And of course, the main product of the Calvin cycle is to produce sugar. Right? So yeah, so if you haven't gotten your notes out yet. If I didn't mention it, I think we're probably going to take our test over cellular respiration and photosynthesis on Monday. So we'll finish kind of notes on photosynthesis today, and we'll actually spend Thursday and Friday reviewing, and we'll take the test on Monday. I don't like doing it on Tuesday because it's an activity period day. I definitely don't want to push it back that far. So we've already spent more time than I would like to on this. So. Yeah, we're not going to skip it. No. All right. So the Calvin cycle happens in the stroma of the chloroplast. So if you remember from yesterday, the stroma is just the liquid inside of the chloroplast that surrounds the thylakoid. So our ATP can go straight from the thylakoid to the enzymes in the Calvin cycle. So this is happening. They're happening right next to each other in space. We also mentioned this yesterday, but we need some of the reactants from the light reactions in order to power the Calvin cycle. So the light reactions are where we kind of are building or transferring or collecting energy, and that energy in the form of ATP in our electron carrier, NADPH, is then what's going to power the Calvin cycle. And after we break down that ATP and we take the hydrogen and electrons for our electron carrier, those are going to go back to the light reactions. So we touched on that a little bit yesterday. So ATP and NADPH are coming from the light reactions. And again, the energy and electrons and hydrogen from those molecules are going to get used to help build our sugar. The product that is not used further for photosynthesis is the oxygen, right? So that's going to be able to leave and go out to the atmosphere. So there's our ATP and our NADPH. So before we actually look at the Calvin cycle itself, there's one very important enzyme that is at the beginning, towards the beginning of the Calvin cycle. And that enzyme, the shortened version of that is called rubisco. So this is the enzyme that fixes carbon. So that takes carbon dioxide gas and fixes it into our organic molecule that eventually is going to become sugar. Here's the long version of that name. Ribulose is phosphate carboxylase. So you just have to know Rubisco. Hint that it's an enzyme is that the name ends in case, right? And this is, I mean, every enzyme is important, but this is potentially, arguably, the most important enzyme in the world because without Rubisco, we couldn't do photosynthesis. Without photosynthesis, most organisms wouldn't be able to survive. So it allows us to capture, carbon dioxide out of air and build all the organic molecules that we need. So because of Rubisco, plants are able to use carbon dioxide to build sugars and then to build lipids and use nucleic acids, proteins, and everything. So this is the basis of the synthesis of most organic molecules. And there are prokaryotes and archaea that are able to get energy and build molecules by using inorganic compounds. But for most life on Earth, this is a very important enzyme. I didn't make this slide. There's a little Kermit the Frog. So that's the important enzyme you need to know about for this section. And this is what the Calvin cycle looks like. You will not be tested on any of the particular products, but I just want you to have kind of a general idea of the main stages that are happening during this process. So these little green boxes here, there's kind of 3 different stages of the Calvin cycle. And we can kind of look at where different molecules come in and where different molecules come out. So you'll notice at the very top of our cycle here, here's our Rubisco enzyme. And that Rubisco enzyme is going to fix carbon dioxide. So this is where carbon dioxide is entering the Calvin cycle. That's really the first stage, is carbon fixation. So we're taking carbon dioxide and we're actually adding it to an already existing molecule. That molecule is going to go through some various changes that require energy. So you can see at this point, here's some ATP entering the cycle. And that's just going to prepare this molecule for further reactions. So stage 1, we're fixing the carbon dioxide, and those processes require some ATP. Stage 2 is reduction. So when we reduce carbon, what do we do to it? We add hydrogen. Okay. So here's where our electron carrier comes in. It's going to donate some of those hydrogens originally from the water, and those are going to get added on to our carbons. So you can see here, here's a carbon connected to 2 oxygens. Now here it has some hydrogen attached to it. So stage 2 is the reduction. At this point, we have our product that leaves the cycle. And so the product that leaves the cycle actually only has 3 carbons in it. And that later, 2 of those 3 carbon molecules can get put together to make glucose. Sorry, I know this is getting really low on the screen. To continue the cycle, most of the molecules have to actually continue in the cycle itself. And so the last last stage of the cycle is just to regenerate our starting molecule, which is this RUBP molecule, and that's going to continue then. Rubisco is going to fix the carbon dioxide to that, and the cycle is going to continue. Okay, so I'm to be completely honest, through both my undergraduate bio classes and some of the graduate classes I've taken, like none of them required me to go into much detail about the Calvin cycle. So I'm definitely not an expert on the Calvin cycle, and I don't expect you to be either. But I do think you can see here, here's the inputs and here's the outputs, and these are the three main stages. So we fix the carbon dioxide, we reduce the carbons, we reset the cycle. And as a result, we can end up making glucose. So we have these 3 carbon carbohydrates that leave the cycle. We can put them together to make glucose. And that glucose can then be used in a couple different ways. So one possibility for that is the glucose can be used to go through cellular respiration, right? So plants have mitochondria. Oh, yeah.
So it's like the glucose sort of byproduct.
Yeah, it's a product of the cycle. Yeah. So really, this molecule is technically the direct product of the cycle, and then we put 2 of them together to get glucose. Yeah. So one option is then we can use that glucose can get transported to other parts of the plant, especially parts of the plant that don't do photosynthesis, and they can use it for cellular respiration and make ATP. So that's one option. What else do you think we could do with the glucose? What are some important molecules that plants need that are built out of glucose? Yeah, I mean, you could really make any organic molecule. Directly, we could make starches. Do you remember what the point of starches are for plants? Energy storage. So we can store it for later. Okay. We can actually make sucrose out of it by connecting it to fructose. We can make cellulose, and we can make all the other organic molecules. So this is a building block to make all the other organic molecules the plant needs. So, lipids, nucleic acids, amino acids, etc. So that is any questions on that before we move on? Okay, again, I think it's important to know kind of inputs and outputs. We're going to focus a little bit more on some math here as far as numbers go. So take a few seconds to look at how much total ATP are we using here and how much NADPH do you see here. So 9 ATP and 6 NADPH. Okay. So we're going to look at a little bit of accounting. So if we go through 3 turns of the Calvin cycle, which is actually what we were looking at on the last slide, you're going to make one of those 3 carbon products. And again, we can put 2 of those products together to make a glucose molecule. So 2 3-carbon molecules can get put together to make a 6-carbon glucose. Oops, wrong way. So in order to get a 3-carbon molecule, how many molecules of carbon dioxide are going to have to go in? 3, because how many atoms of carbon does each carbon dioxide have? 1, right? So you have to have 3 carbon dioxide molecules come in to make that G3P, which has 3 carbons. So to get to 1 molecule of glucose, we have to make 2 of those G3P products. So we actually have to do the Calvin cycle 6 times. So 6 turns of the Calvin cycle to end up getting 1 molecule of glucose. And so that's going to require a total of how many carbon dioxide molecules? 6. 6. And so in total, if we think about what we were looking at in the last slide was 3 turns, we have to double that. So instead of 9 ATP, we use 18. Instead of 6 NADPH to make, we need 12 to make a whole molecule of glucose. Yeah. So the only reason I'm like really going through this is to think about the fact that because we're using sunlight to power this process, we actually only have to put in 18 ATP to get 1 molecule of glucose. If the plant takes that molecule of glucose and goes through cellular respiration, aerobic respiration, it's actually going to end up making a total of around 38, right? And so we actually can generate in the end more ATP if we make the sugar and then break it back down again. So although there's some other processes that go on there. So just to kind of wrap up on the next couple of slides, or just have some, and it is true that if we have leftover ATP from the light reactions, we can use that elsewhere by the cell. But just a reminder that not all cells do photosynthesis. So plants also have mitochondria. They also do the cellular respiration process. And so we're just going to kind of walk through both of those parts of photosynthesis again. So if we look at the light reactions first, this would kind of be a reaction, a chemical reaction for that part of the process. So we have water coming in, we have sunlight coming in, and we end up forming ATP and NADPH and oxygen. There's the water, there's the ATP. So again, our products produce ATP, we produce NADPH. Those two are going to continue on to the Calvin cycle. We also, at that point, release oxygen as a waste product from splitting the water, right? So that oxygen can then leave and go out to the atmosphere. We don't use it any further in photosynthesis. We switch to looking at the Calvin cycle. So here's kind of a overview chemical reaction of just the Calvin cycle. We take in carbon dioxide, we take the ATP and the NADH from the light reactions, and eventually we end up making glucose and other molecules from that. And when we break down that ATP, now we're back to having ADP. And when we release the hydrogen and electrons from our electron carrier, we end up back with NADP+. And so those are going to cycle back to pick up more energy from the light reactions, and so that cycle is going to continue. So if either one of these processes stop, the other one's also going to stop, right? Because there's that cycling of products and reactants. So one of the big questions on the last quick check is filling in this diagram. So you'll have a chance to look and over that yet again. So again, the whole point of the Calvin cycle is to build our sugars, and then we can use those sugars as building blocks for other molecules. We're using that ATP and NADPH from the light reactions, and we're recycling that ADP and NADP plus back to the light reaction so that we can continue to pick up energy there. So just like cellular respiration, you should definitely know what goes in and out of those stages and what goes from one stage to the next. I think that's going to be important for you to know for the test. And Yeah, I feel like this chapter is a lot of memorization. I mean, a lot of this class is memorization. There's also a lot of concepts. So, if we put it all together, here's the total chemical reaction for photosynthesis, right? Carbon dioxide, water, sunlight, yields glucose and oxygen. That's kind of the net total reaction that we've looked at already previously. So there's water coming in for the energy building reactions, for the light reactions. And again, that's where we generate our NADPH and ATP to build the sugars, to release oxygen. We fix carbon dioxide, we reduce it to make our sugars. We recycle that ADP and NADP plus back to the light reaction. So plants, and I don't like saying they make energy. They're really just transferring energy, right? But they generate ATP and NADPH, and they make sugars. So, any questions on that diagram or on how the two stages are related before we kind of wrap up? That's pretty much all the lecture I have. Okay. You have some time then, so you can do both of the last quick checks now, so the 8.5 and 8.6. The 8.5 is really information from yesterday. It's due tonight. I'll start with that one. You haven't done it already. But please use this time to work on those. You can discuss the questions with your group members.
Literally just